guys know what is the meaning of polypharmacology? Anyone? And how does it differ from polypharmacy? Anyone? If you hear the word polypharmacology, what would you guess? Poly means many. Pharmacology means pharmacology, you know. So what many? Very good, right? You are using many drugs. But polypharmacy is using many drugs. Polypharmacology is using one drug which is hitting many targets. That's the difference. Okay? So, you know, for the longest period of time, the key word was magic bullet. That means you have one bullet and it hits the right target. That is the magic bullet. Okay? But now it is the magic shotgun. Okay? So cancer is such a complex disease. If you just kill one protein, it may not be enough. So we have to find all the bad guys and kill all the bad guys without touching any of the good guys. So that is polypharmacology. Is it making sense? So this is what it is showing, that you are hitting many things at once. This one drug is targeting tumor growth, drug resistance and metastasis. No cell phones, sir. See, I teach a lot. I can detect within a second. Like you are doing like this or that, I know exactly where your eyes are, okay? So I teach 600 students class and I can detect who is touching their cell phone. So it's easy for me to see here, very easy, okay? I don't even bring my own cell phone. I mean, here I brought it for picture purposes, but I never use cell phone. I never take it to my class. Okay, so polypharmacology is use of a single drug that targets multiple molecular targets to treat a single disease or multiple disease. Okay, so this approach contrasts with the traditional one drug, one target. For the longest time, when I was a chemist, you guys know, so when I became a chemical biologist, that time the whole world was going magic bullet, magic bullet, magic bullet, one target, one drug, one target, one drug. Doesn't work that way. The diseases are very complex. If you kill one guy, suppose there is a, just think of Steve Jobs, right? There was only one Steve Jobs, but when he passed away, iPhones are still coming because there is always a replacement. So cancer is a living system. You replace one, you target one protein, they will make some other protein, which will compensate sometimes even better than the previous one. Okay, or they will hire 2-3 people to compensate for that one person. So basically the whole rewiring occurs. Okay, so multi-targeting, a single drug molecule is designed to bind and modulate multiple biological targets to achieve a more effective and potentially less toxic. So you can argue, why not we take a combination of drugs? Why do we have to make one drug? Of course, it will be easy to pop up one pill versus 10 pills at a time. But other than that, what could be other advantage of one drug?
Less toxicity.
Less toxicity, and also there could be drug more specificity within the dirtiness, right? And also you are reducing drug-drug interaction. One time, I know of someone. So he had some serious complications, right? So he had to be given many antibiotics at once, and it was just very time sensitive. So I was there to pick up the medicine for this person, and so when so I went to the pharmacy. So the doctors call the pharmacy, and you have to pick the medicine from the pharmacy, right? So when the pharmacist was giving the drug, the system started sounding. This was the first time I heard this was this warning signal, like the police siren kind of thing was going on. So why the system was saying you cannot give these drugs in combination, because if you give this drug in combination, the system, it was 10 years, 2013, so 12 years ago, right? So AI was pretty primitive compared to today, but then even that time the AI said you cannot give this drug combination, this is too toxic. So, but the doctor had prescribed, so I had to pick it up. So I had to sign on the behalf. If there is any toxicity, I'm responsible. Okay, because the pharmacist would not give, because the system was saying you cannot take these many drugs in combination. So polypharmacology means that you actually do whole FDA, whole clinical trial with that one drug and see how it affects the patients in large, like thousands of patients, right? So contrast with polypharmacy. Polypharmacy is different from polypharmacology is different from polypharmacy, which involves a patient taking multiple individual drugs. Okay, so polypharmacology, this is the molecule and it will hit target 1, target 2, target 3. These are all bad targets, but it will not inhibit any good target. So you have to select it against good targets. It should not hit any good target. And then if you take combination, you have slight improvement, but with this one drug, huge improvement. Do you guys know what is synergy? Can anyone explain to me what is synergy? Lekhna, do you know what is synergy? Synergistic molecule?
The activity of the one molecule can be affected by the another molecule.
Yeah, that is, but what is synergy? Yeah, go ahead.
Ma'am, when we give one molecule, like when we do A and B molecules together, then because of giving them together, their activity is beyond, like supramolecular activity we found, like more than their actual activity.
So if this is like 50 effect and this is 50,
Efficiency increases.
Ma'am, the overall effect is more than A plus B effect.
that is the definition of synergy. It is more than A plus B. So if A is 100, B is 100, it is not 200, it is 700. So it's more than additive effect, that is synergistic effect. That is what polypharmacology is aiming to do, highly synergistic. Okay, and adverse effects are none in an ideal situation. So what is the advantage? Improved efficacy, fewer drug interaction because you are not taking combination, simple dosing. You would be very surprised. Remember, she's not here, Shivani, today. Okay, but Shivani was saying it is just really hard to take drugs, like, you cannot take lifelong drugs for something. Thyroxine, right? I am pretty good in this thing, but I've heard compliance is really bad among patients. I know in my extended family, people will not take because I keep telling them these are supplements. I mean, thyroxine is not a supplement, but there are a lot of supplements we take like vitamins and calcium and all those kind of things. I said, what is the problem in taking supplements? Why do you have to leave it? You just keep on taking it. Like you eat food, you just take the medicine. And they don't have any side effect. So, but the truth is that there is a strong compliance issue worldwide. If you have to take more drugs, somebody is forgetting this drug versus that drug, or okay, chhod dete hain, aaj kal le lenge. But it is not good. Some drugs you can leave once in a while, other drugs you cannot. You leave one dose, you go back 1 week because the blood levels go down and it stops working for 1 week till the blood levels come back again. So simple dosing, reduced cost of course, and drug resistance. When you attack some problem through multiple ways, the cancer will not get time to develop a resistance. Okay, so challenges, of course, more polypharmacology, more potential of side effects. Okay, to find a drug which hits all the bad guys and not touch the good guys, it's hard. Okay, as safety risk, and also FDA hates to approve a dirty drug. If it is a clean drug, FDA can approve it. The more dirty the drug is, the more problem it has to get it approved, right? So that is also another problem. So you want to avoid any good targets, you have to make it promiscuous for all the bad guys here. Okay, so one old thinking was one drug, one target. Combination therapy we already talked about, and this is polypharmacology, one drug hitting many things at once. So what is drug combination? You know, right? Drug combination is that you are using more than one drug, and we discussed that in cancer. Remember BRAF and MEK inhibitors together. or EGFR and MET inhibitors. If you inhibit EGFR receptor signaling, it upregulates MET as a resistance mechanism. So if you give both, you kill them, right? So this is all still going on. This is HIV combination therapy, which use 3 antiviral drugs, 3 HIV and 3 retroviral drugs at once. Okay, the best. So this is still going on, and this will still go on. This is not going to go away. Combination therapy will keep going on, especially for complex diseases. Even right when I was telling cancer patients are given anti-angiogenic therapy plus PD-L1 therapy, and that is extremely effective. Okay, but today I'm going to tell you about molecules which can do both things. One thing, but capable of doing anti-angiogenesis and anti-proliferation. Okay, so this is just comparing combination therapy with polypharmacology. This is cocktail of drugs, many drugs like BRAF inhibitor, MEK inhibitor, multiple chemical entities, the risk of drug-drug interaction, complex pharmacokinetics. One drug has a short half-life, other drug has a long half-life, which one to take first, which one to take later. Sometimes you are supposed to take drug A at 6 a.m. but drug B at 6 p.m. and maybe you have to take drug A twice a day, drug B once a day. So it's pretty complex regimen. Okay, and higher pill burden. Polypharmacology, one drug, many targets, single chemical entity. Because you have one drug, one molecule, low manufacturing cost. Okay, so this is the paradigm shift. So when I started my postdoc, the whole world was going after magic bullet, that one bullet hits one target in the cell. Now we are going for magic shotgun. It is hitting all the bad guys at once, and that is how we can overcome resistance. Okay, so this is the guy who gave the term Paul Ehrlich magic bullet. He's a Nobel laureate. Okay, he was a Nobel laureate. If he gave it, I don't know about when he coined it. He may be still around because magic bullet came in 1990s or 2000. So it's a pretty new word. Okay, and now we use magic drug. This is single target, this is multi-target. Okay, you guys already know about this thing. So for schizophrenia, there is a drug. Okay, and that is so surprising. So there are so many drugs. There are so many people who are suffering from that. Have you heard of any person who has that, even from distance? No. So one of my student, who was my PhD student, developed it. He was the smartest guy. I have never seen smarter kid than him. 10-15 years after he graduated from my lab, now he has those symptoms. People who are super smart, and if the energy is not directed towards the right direction, they may have some propensity to develop it. I don't know. But now there are drugs which inhibits many targets, and it is very effective for this disease. Okay. So you guys already know what is. So I still included this picture. This is really good. We have published many papers where we have used indole ring and modified it all around it. So our collaborator Bits Pilani, he made those compounds and we just tested them. But the beauty is that, look at the indole ring. Many of you are chemists here. So look at the indole ring. So if you substitute second position, fifth position, this will become anti-asthmatic drug. If you substitute fourth position here, it will become cardiovascular. Just see, just by changing the modification, you can target different kinds of things. So now if you have to do polypharmacology, you know exactly what to do, right? So why single target drug fails? Because so suppose I am very important, and if I leave, somebody else will replace me, right, as a teacher. If there is a CEO of a company who is extremely important, Bill Gates left Microsoft, it is doing extremely well, right? The CEO is someone else. So the cells are living. You remove one target, they will overexpress something else. So compensation, okay, so solution is that you attack multiple things. So the point is to generate precisely dirty drugs. You are making dirty drugs. but right amount of dirtiness. Okay. So if target receptor is A, B, C, but not target D, E, F. Okay. So synergistic is more than 1 plus 1. Okay. So this is the formula. So if you check it out, then you can. Have you ever worked with a synergistic, like you add two drugs and they have a synergistic impact? Have you done that? You will. If you use drug in combination against cancer cells, there will be drug combination which will be highly synergistic. Okay. So how do you do polypharmacology? How do you do it? Many ways to do it. But one way is repurposing of drug. How many of you have heard of repurposing of drug? What does that mean?
Like one drug is already approved for a particular disease, and now you are using the same drug to target to treat another disease.
That is very good. Repurposing. So repurposing became such a key word or buzzword 10 years ago. So the most important example of this is metformin. So how many of you have heard of metformin? And this is for?
The anti-diabetic.
Diabetic, right? What is the commercial name here? Because my uncle was telling me the commercial name for metformin and I said, I don't know. What is it called here? Is it called metformin? You guys are scientists, so you know metformin, but the commercial name is something else for metformin in India. And if you hear that word, all of you will recognize that name. But the point here is that this is one drug which is like a supplement, which is good for you. Okay, this is given to diabetes patient. It is also given to pre-diabetic patients who have a chance of developing diabetes, right? So metformin, what is the side effect of metformin? It is anti-cancer. How good? How good? You are pre-diabetic, so you are not going to develop diabetes, hopefully, if you keep control at your sugar level, but it is anti-cancer. It's really awesome, right? So what is the advantage of repurposing a drug? If you start a drug from the scratch, it's a very long process, like preclinical, for clinical 1, clinical 2, clinical 3, but metformin has already passed everything for diabetes. So now you have to check the cancer, so you just go right up here at the final stage and see that. So it cuts the cost tremendously. Keep in mind, when a drug is developed, whether anywhere in the world, right, the drug is very expensive initially. Why? It is not the actual synthesis of the molecule. That may take 1,000 rupees altogether, or even 500 rupees just to make the molecule, correct? But it is the discovery process of 10 years which led to that. So the company has to recoup all that cost, and that is why the drug is very, very expensive initially. Okay, so but if you repurpose, you save tremendous amount of money. Okay. So polypharmacology. So I'm going to tell you many things. So polypharmacology it hits many kinds of different drugs. I have to tell you a joke, right? So I was making these presentations tomorrow. I had made it before I came here, but I reworked on it. So I don't have any of the AI programs, so I use the free AI programs. So ChatGPT doesn't let you make more than few figures, right? So it was funny. Yesterday I was making figures after figures after figures, and it wasn't stopping me. So I thought maybe on Christmas Day they've opened it infinitely. Which is very few figures are there, but for fun I made like 20 figures and didn't stop me. So I said two of the one things happened. I'm the only one working on a Christmas Day on ChatGPT. So it's allow me, or it is a Christmas present. And they said whoever. After 2025, it said, okay, finally stop it. You have hit your limit for today. But it allowed me many more figures than a normal day. But this is one of those figures which I made yesterday. Okay. You can always find out an AI figure because it will have spelling mistake. Okay. Invariably spelling mistake. So combination therapy, network, I will come to all of these things instead of telling you here. So polypharmacology can be achieved in many different ways, and these are some of the ways. Okay, so okay, just 1 second, something is wrong here. Just 1 second. Yeah, you know, something is wrong here. I accidentally put the wrong thing here. There were many of them here. Where I will go home and I'll correct it before I send it to you guys. Let's stop here. There are many ways to achieve polypharmacology. I spent 7 hours doing it. It is not that. Somehow I put the wrong slide here. So now I'm going to tell you few ways by which people are able to make drugs which can attack multiple targets. So sometimes what you do, you just have a simple one drug and it just is a magic drug. So when Gleevec was discovered, the truth is that for Gleevec, so Gleevec was developed by Novartis and it was developed against BCR-ABL. And when this drug was marketed, right, so that time nobody knew that it is such a magical drug in the sense that they had made it against BCR-ABL, but it also inhibited c-KIT and FLT3 receptors, which are tyrosine kinases. And they are also upregulated in CML. And Gleevec is against CML disease, chronic myeloid leukemia. So it just happened to be the magical drug which was hitting all the right targets. Okay. So later on people made a drug which was against pure BCR-ABL and that wasn't as effective. So the dirtier drug Gleevec was more effective, and that is when people realize you have to hit more than one target. So imatinib or Gleevec hits BCR-ABL, KIT, PDGFR, although it was developed against BCR-ABL alone. But later on when people realized, yeah, hitting more than one target is really good. Now we intentionally make drug which can hit more than one target, okay? And sorafenib is one of them. So again, the pros are it is simple dosing, lower cost, but then you have to choose your targets very efficiently, okay? So one of the ways to make a drug which will hit multiple targets is to do network-based polypharmacology. So you are not going after one target, you are going after one whole pathway or network. So suppose, so this is not the best picture here, but what it is showing here is that this is EGFR receptor, this is MAPK pathway, this is mTOR pathway, NF-kappa B pathway. So they are all connected. Signaling pathways are not linear, like they are going like this, but they are also going like this, they are going in network. So suppose you know that in this patient, this pathway is regulated, whole RAS pathway is upregulated because of KRAS. You make an inhibitor for KRAS mutant, but you also target downstream along with that. So you can actually, or suppose two things are talking three things, so you can make a drug which will hit the main nodes, okay, critical hubs. If you hit those, then you have a better chance of killing the disease, okay. So that's what it is that you can, you know, these protein-protein interaction networks, you can, I mean, people have been making it for decades now. They were not very good before, but now they are very good, because what happens is that all of us, right, all of us worldwide, when we do gene chip analysis or RNA-seq or different kinds of network analysis, now everything is, journals will say you have to submit the raw data, right? So basically everybody has access to all the data and they can analyze the data. So it's becoming a big database of all the data. So now AI is also becoming smart. The richer the database, more things you can analyze, correct? So protein-protein interaction networks, gene regulatory networks, they are becoming more and more sophisticated. So you can say, okay, this person, we have to shut down this whole pathway here. So you can use proper combination. So a very simple example is simultaneous inhibition of PI3K, AKT, mTOR signaling. So you guys don't know what they are. So what happens is that, look at this thing. This is receptor tyrosine kinase. So signal goes here. It goes to PI3K. From PI3K, it goes to AKT. From AKT, it goes to many, including mTOR. So instead of inhibiting only PI3K or only AKT or only mTOR, what if you make a drug and you inhibit all three? So no signal can leak out. You are just inhibiting completely. So this is a drug, vertical pathway. This is a vertical pathway, you are hitting all three. You make a compound which hits all three. Yesterday I spent a lot of time so that I can find a figure where they show the whole molecule like this, and they say this part inhibits PI3K, this part inhibits mTOR, this part inhibits AKT. I could not find, because I was hoping for some color imaging, like red is AKT, one molecule. So suppose it is one molecule, this part is hitting this part of the kinase, that I could not find it because it may not be that simple, or I was just not able to search it properly. But the point here is that you can make a molecule which can hit all three of them. But here is the challenge. The more complicated the molecule, the more side effect it will have, because it may hit some other targets also, right? So, but the idea is that you can vertically inhibit the thing, like all three of them, or single molecule dual inhibitors, like you have actually one molecule but it is hitting several of them. Drug combination is not polypharmacology, and PROTACs can be used along with small molecule. So you can make a PROTAC. What is a PROTAC? Can you tell me? Targeted degradation. So you have one part which will identify protein of interest and one part will identify E3 ligase, right? So this molecule which identifies protein of interest can actually bind to more than one protein and degrade more than one protein. That would be polypharmacology, right? So old paradigm was one drug, one target, one disease. Now multi-target, you are hitting the whole network of proteins which are involved in that, and for one. Now there is another way to do that, and that is called fragment-based and scaffold hopping approaches. So what you do is that, how many of you know of fragment-based chemistry when you make molecules? Some idea. Can you try to explain to us? Let me explain to. So fragment-based, what is fragment-based drug discovery?
Are you talking about retrosynthesis?
No, not retrosynthesis. Fragment-based drug discovery.
Collect different, like we have already online available drug repositories.
Yeah.
And it helps us to develop different fragments. By combining them fragments, we finally build one active molecule.
Exactly. So what you can do is that, you have many different fragments, like many different chemicals, right? So these are all different kinds of fragment, A to Z, let's say 1 to 100, and here you have other fragments 101 to 200. You can combine them in every possible combination, two fragments, right? So you will develop a whole new bunch of compounds. That is, in very simple words, that is fragment-based discovery. Then what happens, you say, okay, compound number 12 here in fragment 1 binds AKT, compound number 22 binds mTOR. You combine them together. Can you inhibit both kinases together? Is it making sense? So, and also, see, the beauty of fragment-based drug discovery is that suppose you have 100 fragments here, 100 fragments here. Now you just have to clip them together. That is much easier. And you think about it, all permutation, you will make thousands of compounds, right? But if you have to build each and every compound, how many syntheses that would be? Here you have made large scale 100 compounds, large scale 100 compounds. And you can build thousands of compounds just by clicking it. You can even use click chemistry there, right? So the point here is that fragment-based drug discovery is a very powerful way for building up libraries of compound. So that's what it says, fragment-based, and I will tell you what is the meaning of scaffold hopping approaches. So this is just a very simplified version. You take fragment A, fragment C, and you couple them together and you can make large molecules. This is actually a drug here. So this is fragment A, this is fragment B. This is a FDA approved drug. Okay, so this is fragment B, and you are linking them together, and this hybrid molecule targets CSF1 receptor. Okay, now let me tell you what is scaffold hopping. So this is a natural compound, orones. Okay, now they have seen instead of oxygen here, if you replace it with NH here, this is better. So if you do this, there is higher chemical stability. Now instead of N, you also introduce an N here. Okay, so it's imidazole like structure. So this one, you gain all these properties. If you change orones to these things, here you are putting sulfur here. So this is scaffold hopping. The basic structure is the still 5-membered ring, but just by replacing with some heteroatoms, you are changing, you are making them highly stable, higher cell permeability, higher oral bioavailability, better pharmacokinetic properties, less target promiscuity and off-target interactions. So that is called scaffold hopping. Okay, so now how do you do the fragment-based drug discovery? So let's say these are the two fragments, A and B, right? So you can attach them by a linker. So A is there, B is there, there's a distance between them, that is linker, right? That is linker based. Or you can fuse them together. So A is still there, B is still there, but there is no linker, they are fused together. Or you can merge them together, okay? Like Ardhanarishwar, okay? Like half of this, half of that, or whatever, right? So it's that. So that is what it is. So this is linker, this is fused, and this is merged. So you can make all sorts of compounds, or you can make a compound which is not a merged, fused, or linker, but that compound itself hits many targets. Okay. So here is an example. See here, I'm just trying to find out which one is linker based and which one is. So this one is linker based. This is one part, this is one part, this is the proline like structure is the link. It's not proline like, but this is the guy which is the linker here. In this, dopamine and this pharmacophore are attached to form this compound. Okay, here is yet another compound, two pieces. I have some better examples here. Sunitinib, imatinib. Imatinib is actually not it was not developed as that compound, it just turned out to be that. But look at these compounds here, okay. This is just a linker, this is a fused drug here, okay. This is merged drug. I have better examples of merged drug. Here I will show you, yeah. So just a little bit more about fragment based and scaffold hopping. So this drug is a PI3K inhibitor plus mTORC inhibitor. I could not find a very nice picture of this one which will differentiate the two, but I made this picture. This is obviously incorrect because hydrogen cannot form a double bond like this. It is a nitrogen there, okay. But this is showing you a hybrid drug. One part is a cancer kinase inhibitor, one part is a HDAC inhibitor attached with a linker. This is obviously wrong drug. I mean, the molecule is itself wrong. But many compounds are dual PI3K-mTOR inhibitors or HDAC kinase inhibitors, just like a cartoon like there. True merged pharmaco, not a combo. So look at this. These are very good examples. So this is one part of the drug. This is paracetamol. Paracetamol is for what? Anti-inflammatory, antipyretic, right? So you mix them together, okay, and now you see the whole of it. So this is fusion or merged, or what kind of drug is that? The first one. Fused, because you are not really removing any part. So this is fused. These two together, this is really beautiful example of merged, right? Because this ring is actually becoming this ring. That's why they have changed it green. That's what I was looking for it yesterday. This is really beautiful. This is a perfect example of a merged drug, works both ways. And this guy here, these three are together to make this, of course, merged, right? So you can see the examples of these drugs here, right? And molecular docking is very helpful in that case. So when you are developing those drugs, how many of you have used AlphaFold? And the AlphaFold is different, but have you used AlphaFold? AlphaFold tells you about the protein structure, and you can take that structure and also do docking on top of it. But many of you do docking, right?
Yeah.
So yes, so docking is very, very useful. Okay. Now let's go to multiple target biologics. Something is missing here. This is number 5. Number 4 is missing here. I will correct it once I get back there. So again, you can use biologics to target many things. It's relatively simple here. You can make a bispecific antibody. One is targeting the cancer cell, one is targeting the immune cell. Bring it close. It will kill it. Here, this is a tri-specific antibody. It binds 2 different antigens and a tumor cell here. Okay, and this part will bring NK cells, right? I wanted to include this one to show you something very interesting. So I told you up to the nanobodies, right? So antibodies are 150 kilodalton, then there are dye bodies. This course doesn't have enough time to go over everything, but then dye body, this is smaller, then FAB 55 kilodalton, then SCFV. Have you heard of SCFV, anyone? I hope there is one person who has heard of SCFV. Where did you hear about it?
In tandem CAR T therapy.
CAR T, not tandem, all CAR T have SCFV. Please remember that all CAR Ts have SCFV. The thing which recognizes the antigen, it's not the antibody, it is SCFV. Okay, so it's much smaller. How small compared to antibody? So this is CAR T. Then we never talked about any of this except nanobody. So anti-calin, nanobody. Look at this thing. Nautin is just a tiny bit. There are 3 sulfide, disulfide bonds which stabilize the structure. This is very stable. Just 4 kilodaltons, this thing. Okay, so we are coming back to fusion proteins. So remember I told you, you can have bispecific, trispecific and fusion proteins. So these are fusion proteins. So monospecific, right? One binds, this is a protein of interest. This is your one compound which will bind the same. This is multispecific, binding 2 different proteins. If it is multispecific, see receptors on the same surface. This guy can bind receptors on the same surface or receptors on different cells or different binding sites, like this part is binding here, this part is binding here. It can bind the ligand. So you can have all different kinds of combination. Okay. Now let's do PROTAC based polypharmacology. So what are PROTACs? Remember about PROTACs, these are very important, right? What advantage PROTACs give to you as compared to small molecules? Yeah. Anyone? Why would you use PROTAC and not a small molecule? Go ahead. You can target the undruggable thing. Yeah, because it is hard to make PROTAC for everything, right? Small molecule for what else? You can make a small molecule with dual target too. Degrade the protein. See, small molecule, imagine I am the whole target here, right? So if you are inhibiting the thing, you are just putting something in the mouth like big rasgulla, which cannot be eaten, right? So you cannot hear the voice, but the whole thing is there, hand gestures are going on. But if the protein is doing a lot more than just the enzymatic activity, and it is doing a lot of interaction, making it disappear is very useful, right? So you degrade the whole protein. So that's why. So you can also do PROTAC based polypharmacology. You can make one PROTAC which can degrade many targets. Okay. So for example, you can make dual target of PROTAC which will inhibit CDKs and an oncogene. So look at this one. This one here, this target will inhibit 2 different targets, synuclein and tau. Okay, look at this PROTAC here. So this PROTAC is inhibiting one protein of interest and also the second protein of interest. So basically, the molecule which is there, which is binding more than one protein, and no matter which protein it binds, it will degrade it. So it is dual specific, or you can make two different PROTACs for two different proteins, right? They are attached to the same E3 ligand. Is it making sense to you? Keep in mind, you guys have your exam tomorrow, so pay attention to the details, okay? When I'm asking you questions, that means these are my favorite questions, okay? When I teach a class there, I always tell them, because no matter, even if you're teaching a semester-long course, you tell a lot of things. Some of the things are not important from exam point of view. In my time, professors would never tell us what is important for the exam or not, and we could never dare to ask. Anything which is discussed in the class could be in the exam. But nowadays students want to know, and I tend to also say that I'm including some slides just for your knowledge. It's not going to be in the exam, right? Because when you show slides, sometimes it has more information than what you want to give it to the students. So I always tell them that if I say something is important, that means it's going to be in the exam. That holds true here too. So if I'm asking you about certain things, pay attention to those things. Okay, so you can see that, right? You can build PROTACs which can attack a lot of family members altogether. So this is one compound. See, this is really good. This is the part which will inhibit two kinases at once, CDK5 and GSK3 beta. Okay, so this is a dual PROTAC, and this is the one which will bring E3 ligase. Okay, look at this thing. It's just really good compound. Okay, at 42 nanomolar, it will inhibit GSK3 beta and CDK5. Then you can also do polypharmacology by hitting something. There is some kind of toxicity issues also, but suppose you hit heat shock proteins. What are the functions of heat shock proteins? Do you guys know? Have you guys heard of heat shock proteins? Tell me, Shivani.
I don't remember them completely, but they were like something to do with
Conformation. So what heat shock proteins do, these are chaperone proteins, you're right, and they help in protein folding, especially under heat shock conditions. And also they help in the protein folding of many different proteins. So sometimes when you inhibit a heat shock protein, they have many client proteins. That means one heat shock protein may take care of 20 different proteins folding. And if these 20 proteins are all bad proteins, so most of them are bad proteins, you hit one heat shock protein and all the other proteins will not fold properly. And if they are not folded properly, they will all be cleaned by the proteasome system. You know, when the proteins are not folded properly, they are degraded, correct? Because the cells will recognize it. So this is an extremely dirty polypharmacology, very, very dirty, because some good things will also not get folded, right?
This will have very high off-target toxicity.
It will have very high off-target effect. You are correct. So this is like not the method of choice, but this is just to show you. Just whenever you see so many of them, it is scary. Okay, but this is also one way that you are just shutting down the whole pathway just by hitting one protein. Okay. Now this is extremely useful, this one.
heat shock proteins were those when we were discussing about transfection and plasmid incorporation.
So that's you give heat shocks for 30 seconds to 45 seconds. So that is not enough to give a heat shock. I mean, they will take the plasmid in, but because what you do is that you keep the cells, these are not rapidly going cells, they are frozen cells. So you put them in ice for 30 minutes, so nothing is going on, they are not dividing at 30 minutes on ice. Then you give heat shock, so they just suddenly take the DNA in because they were on ice for 30 minutes, then you are putting at 42 degrees, then you put them back on ice for 30 minutes, no, 2 minutes, and then you grow them. So by that time the heat shock thing is gone. But what I was telling you about heat shock is that if you take E. coli, which is growing at 37 degrees, and then you put them at 42 degrees, then they will overexpress heat shock proteins to prevent the proteins from getting denatured. But these heat shock proteins are naturally expressing in the cells to aid in protein folding. These are not necessarily induced by heat shock in humans. These are human proteins, right?
Degrade the proteins which are not properly folded.
No, they are not degrading. It's the cellular damage control machinery is doing that. So when a lot of complex proteins are produced, heat shock proteins help in protein folding. Okay, yeah.
If you have to like gist it up, like what is the gist of the whole thing, like heat shock proteins? If you have to know it in like few lines.
So few lines it is this, that heat shock proteins are chaperone proteins which help in protein folding. Okay, that's the main thing. In E. coli it has a different function altogether, because some heat shock proteins are produced when you put E. coli growing cells in under non-physiological higher temperature. So then these proteins are produced to help to prevent other proteins from getting denatured. Make sense? Yes. No. Heat shock proteins are produced across the board from E. coli to humans. What happens is that, so here is something, I'll tell you something very interesting. So as RNA is making, as ribosome is making the protein from RNA, right? So as the protein peptide is coming out of the RNA, it slowly gets folded, okay, and the whole protein is folded and final protein is made. If there is some mutation in the RNA itself, then the protein will not fold properly, because the protein will also have the mutation, correct? So then, and if it doesn't fold properly, it will aggregate, and the cellular machinery will clean up the aggregate machinery. It's like trash. So trash is cleaned, right? So there should be no aggregation. So that is the way, if RNA is messed up, the protein will be messed up and they are completely cleaned. So we don't have any damaging effect. But if the DNA is messed up, it's always there. So then it is a problem. That's why people say genetics is messed up, that can give rise to disease. RNA messing up doesn't give rise to a disease unless it is a microRNA. Okay. So heat shock proteins are needed for some proteins to fold them properly. So as they are coming out, they still need assistance from heat shock proteins to fold properly. So in cancer it is shown that many oncogenes, these oncogenes are not natural proteins, right? They have deleted some part, they become hyperactive, they have undergone mutations. So they need assistance from heat shock proteins in properly folding. So the idea is that if you inhibit heat shock protein, you are inhibiting many proteins at once. The stability of many proteins at once. And if they are not folded properly, they will all be cleaned up. Okay. Is it making sense? Yeah. Okay. I will tell you something very interesting here too. Do you guys know about codons? Have you studied genetic code? All of you? Every single one of you? Have you guys heard of codon optimization? No. Okay, so you know, can you name any one amino acid which has 6 codons in the genetic code? So methionine has 1 codon, AUG only, or ATG, whatever, right? It has only 1 codon. Tryptophan has only 1 codon. Glycine has 4 codons. Alanine has 4 codons. Leucine has 6 codons. Arginine has 6 codons. Okay? Why I know all these things? Because I teach this thing. Okay? So I will tell you a very simple example, and that will tell you something very interesting. It was a science paper. Okay? So glycine has 4 codons: GGA, GGC, GGU, and GGG. Okay, 4 codons. So let's say in humans, Not all 4 codons are used. Let's say, for example, for humans, GGG is the predominant one. 90% proteins, if they have to make glycine, they will use GGG and rarely GGA. Let's say, this is hypothetical part. Codons are correct, but which one is used more, that I don't remember. Okay, but in E. coli, GGA will be used. Okay, so if I'm making a human protein in E. coli, okay, so human gene has GGG because humans like to use GGG for making glycine, but E. coli likes to use GGA, correct? So what people do, you pay a company and you say that you codon optimize it for E. coli. So it's a human protein. So human protein will have GGG, for example. So it will say change it to, human can also use GGA. Remember, it's a universal code. We have the same code as E. coli. But because E. coli have more tRNAs for GGA than GGG, they will say, okay, optimize it so that E. coli can make the human protein efficiently. Once the protein is made, GGA, GGG doesn't matter, right? Because it will be glycine. Do you guys understand what I'm trying to say? You guys are able to understand? Should I repeat it? No, you got it, right? All of you. So, some scientists were doing very simple experiment, thinking of publishing the paper in 2 impact journal, right? They were just trying to make a protein. They just give the company some money to codon optimize it. The company optimized it and then they express it in E. coli, and the protein never formed. It would just form aggregate and just crash out. So they could not understand. So then they went back to human cells and then they optimized it perfectly for human also. So what they did is that wherever they had GGA for human, they made it GGG, thinking that the protein will be expressed better because humans like to have GGG, but the DNA sequence had GGA at some places. So they changed it to GGG. It would not express in humans also. It was just degraded. The moment it was expressed, it was degraded. Can you explain to me why? So let me summarize the problem. So there is a DNA sequence, and let's say there are 10 glycines. And humans, we know from before that humans like to have GGG for glycine. But in 10 places, 8 places had GGA, which is not liked by humans to express glycine. So these guys made all GGG, all 10 of them, and the protein would not express. And we know that humans have tRNAs for GGG more than GGA.
Does it need C because it is triple bonded with C? Does it have something to do with stability?
Codon has nothing to do with triple bond because it's not double stranded, it is just single stranded, so there is no hydrogen bonding that way. So it's not that. So think bigger, because like I said, this paper would have come in a 2 impact factor journal. It came out in Science because it was such a basic thing which was going wrong there. So I want you to think big picture. Think of it as if you are witnessing the whole drama in front of your eyes. So let me repeat it once again because I want all of you to think about it. So because you guys are scientists, you are in research, you have to, if something so strange comes out, so suppose you had all GGG wherever glycine was there. tRNAs are the one which bring the amino acids. So if you have better tRNAs for some codon, it will be faster synthesis because you have ready supply, right? So the parental DNA which you isolate from the organism at 8 places, let's say they had GGA, which is a bad codon for glycine for humans. And they were making it in human cells. So they gave it to the company to change it to GGG, which is considered a good codon for human. But the protein would not express. It would express from the bad codon. Like if they use the original DNA, the protein would be made, no problem. But when they were using this codon optimized one, which is supposed to be better, the protein would not express. Same cell line.
Because it's coming from the DNA.
No mutations. we have introduced mutation, but these are functionally silent mutation, right? Glycine is encoding for glycine. So you cannot even say that the protein, you have introduced a new amino acid and protein is not folding properly. You cannot say that because glycine remains glycine. You are just changing the codon.
Because of introns and exons.
No, it is a mature, when you do the plasmid, introns, exons don't come into picture. It's just the DNA directly making the RNA.
Ma'am, maybe it's because coming from the mother DNA which have GGA. Is it because
Why that is making a difference?
Is it their identification that if this proportion they have?
No, what identification? No, so think about it a little. I want you, like when you are in the jungle, right? You are looking at one or two trees, but if you go up, you can see many trees, and you go up like a bird's view, you can see the whole jungle. So I want you to come out of that thing and see what's going on. Tell me.
Any other codon which was coding the protein that helps in folding was specific to GGA?
No, so GGA and GGG, they are both glycine. So suppose I make from GGA, I make GCA, then I'm changing glycine to alanine. Then of course it will change the protein folding because you are putting a new amino acid there. But the thing is that they have not even changed the amino acid, they have just changed the DNA sequence.
Ma'am, if I'm not wrong, the problem is you have a continuous strand of GGG for the 10th.
No, it is not continuous strand, it is a long DNA, and let's say it has 10 glycine. I don't know what was the exact issue in that protein, but let's say it has 10 glycine. So at some places they had GGC, which is not such a good codon for humans, and the answer is hidden in this sentence, which is not such a good codon for humans. They replace it with GGG, which is a very good codon for humans. So tell me, I'm going to guide you to the answer. Go ahead.
Maybe because in like it may be interacting with some other.
Can you, if you speak the right answer, I want to have you this thing.
Maybe it will interact with some other amino acids of the DNA. Let's suppose if it is like at 50th position, so it's not like it will interact with only the nearest one. Maybe when protein fold, it may interact with some other, like maybe 70, 80.
No, you are right in that respect, but the thing is that glycine will still remain glycine. So 50th position was GGC, right? And we changed it to GGG. But it is still glycine. When
Yes,
it
ma'am.
was GGC, it was glycine. When it is GGG, it is glycine. So if it was interacting with the 70th position, it will still interact with the 70th position. You are going in the right track. You have to just go a little bit
Yes.
further.
I think it is coding for glycine only
Yeah.
right now also. But I think it's the interaction, like the interaction which are going on inside that. Like maybe A will interact somehow differently with other amino acids which are present, and it will determine the aggregation of that. Like it may be degraded, like the protein. Like the
So
place where
if
G
the-
was supposed to be, there is A now.
No. That's what I'm saying. You are not changing the protein sequence at all. You are not changing. It is identical. Okay, so I will give you a clue because I won't give you the answer. I will keep on giving you the clue till you guys go. Just give me. Yeah, go ahead.
Do they form dimers?
No, they don't form dimers.
Does it have something to do with GGA when it's about folding?
GGA will always bring glycine and GGG will also bring glycine. You guys are coming very close. You are almost at the edge of the door, but you are not getting the whole picture. So, okay, now I'll give you a clue and all of you will get it. So what happens if you use a poor codon? What will happen? It will still encode for glycine, no doubt about it, right? Because it is glycine. But what will happen?
In
Functionality.
functionality.
No. As protein synthesis is going on, you guys know how things work, right? So tRNA has to be there. But if there are not enough tRNAs for GGC and there are too many tRNAs for GGG, so anytime GGG comes, protein synthesis will be faster. Anytime GGC will come, protein synthesis will be So nature has made it slower at that point because that was an important junction where it needed to slow down for proper protein folding. Okay, sequence is the same. So it had to slow down. When we were putting GGG, it was just so fast. The whole thing was aggregating. It was getting cleaned up. Protein was degraded. So nature has chosen for it to slow down there by using the worst codon. And when these guys did codon optimization, that's how it became a science paper. It was such a new discovery, and it was a recent science paper, like 4 or 5 years ago. So just imagine slowing down also at places. Suppose you're driving a car and you're going over the hill where there's a real danger, you have to slow down. So proteins folding at that juncture was so important, it needed to slow down. Okay, so my point here is that if you see something so unexpected, right, when you're doing the good thing and something bad is happening, nature is telling you something very important. So look at the big picture, step out of the problem. Somebody I met and we were having dinner together, she's the Director General of something of cybersecurity. And she lives in Bangalore, which is a very nice city, and she was posted in Delhi for 4 years. So the first question I asked, so how is the pollution? And she paused for a minute and she said, when you are within the problem, you don't see the problem, right? So when you're too deep in that problem, you are not thinking about protein folding. So you need to step out of the problem and see what is going on. Let me see. Okay, so that is there. And now you are watching, observer, right? Have you heard of the witness thing? You witness the thing and don't become part of the story. That's the witness thing, right? As a scientist, you just witness what is going on there. Okay, so now you guys know what it is. We had a fun discussion, right? Okay, let's go to the main story here. This is very interesting. Phenotype-driven polypharmacology. How many of you have heard of forward chemical genetics versus reverse chemical genetics? Nobody has heard of it. This was a very popular term in the late 1990s. So what happens is that there are two ways to drug discovery. You say, oh, BCR-ABL is there, and now I'm going to make BCR-ABL protein pure, and I'm going to screen millions of compounds against this pure protein. So the goal is to inhibit BCR-ABL. So that is called reverse chemical genetics. You start with a target, and then you screen millions of compounds. Forward chemical genetics is that I don't know the target, but I want to kill the cancer cells. So I take cancer cells and I screen millions of compounds, and then I choose those compounds which kill the cancer cells, and then I take those compounds and I use it against normal cells, and I want normal cells not to be killed. So then some will kill both normal and cancer cells, we throw them out. But those compounds which just kill BCR-ABL cancer cells or any cancer cell and not normal cells, they are my hit. That is called forward chemical genetics. Okay, forward chemical genetics is phenotype. Okay, I want phenotypic change, live, dead. Like this kind of cell versus this kind of cell, that is change in phenotype. Phenotype is how we look, right? Our DNAs are 99.99999% same, right? Anywhere in the world, if you compare to any human being, right from somebody in Antarctica, our DNA to the DNA of those people, 99.9% identical. Phenotype, entirely different, even in this small room, right? So DNA is same, phenotype is different, right? So when you are doing phenotypic screen, you select drugs by outcome, not known targets. So then you can understand polypharmacology. I want to kill cancer cells. If it is hitting 10 targets, 20 targets, I don't care, as long as I am killing cancer cells. So that is the most important thing, phenotypic driven polypharmacology. Okay, I'll come to that in just a few minutes again. Like she said, Janvi, drug repurposing, that is, so this excuse me, target this for one disease, some drug was identified, okay, but we also said, okay, in this disease it is hitting this target, but this is the side effect. But if the side effect target C is cause for another disease, you can use that for that disease. So look at this thing, how long it takes for the drug clinical trials. You do basic study, you do drug design, you do in vitro preclinical studies, then phase 1, phase 2, phase 3, and then the whole thing. But if you are taking a FDA approved drug and repurposing, it takes really a short time. Sometimes, in COVID time, the pressure was so high, people never thought of mRNA vaccine, but people were dying across everywhere in the world. It was just not ending. It was just unbelievable everywhere. Like, people are very nice and very considerate under better circumstances, but when things go a little bit haywire, like everybody was breaking every rule, right? So we were under tremendous pressure to find a drug, and so we made the discovery. We would have never made it at that time. Like, there was so much pressure, right? So at that time also, AI helped in drug repurposing, taking all the FDA approved drug. Like every lab was working on COVID at that time, just trying to do their part. Okay, so success story, of course, metformin is one of the best story. There was this thalidomide thing which were given to pregnant women, right? Now it is given to multiple myeloma patients. Okay, then there is another thing called chemogenomics in polypharmacology. So here what you do, you take a whole compound library, one compound at a time, and then you add it to the cells and you do the whole pathway analysis like genomics, proteomics, metabolomics, RNA-seq, everything to see exactly what are the different things it is hitting. Like, you get the whole profile of it, and then you can plan which drug can be used for drugs. But the thing is that this gives you so much information that it is hard to like make sense of this much in this thing. So that's where artificial intelligence really comes into picture. Okay, that is what we will see in deep medicine. So this is like a whole library of compounds. You can give genetic treatments, you can give small molecule treatments, and then you can look at many different, and you can build up network, and then you can do that. And then finally we'll come to AI and data-driven approaches. So our 30th lecture is all about AI and data-driven approaches. So when we started the lecture, we were talking about conventional therapies for cancer, like chemotherapy, right? Chemotherapy, surgery. Then we went to precision medicine. And AI is beyond precision medicine. AI is that, will not let the disease develop. Precision medicine will only come once the disease develops. So AI is supposed to be proactive and telling you years in advance that there is this risk factor and the disease never develops. Okay, so that will come. So AI and data-driven approaches. So drug target interaction prediction, it can do multi-target design and optimization. It can repurpose existing drugs, predicting adverse effect. So many things at once it can do. Okay, and then you can find exactly the right compounds, and AI can even dream up molecules and even do the synthesis of molecules. Have you seen or have you heard of labs where people are doing synthesis all by robotics? Anyone in IIT Indore?
People in China do that. So Venkatesh was telling us that.
Yeah, but I went in 2016 or 2019. I think in 2016 I went to IISER Bhopal and IISER Pune and IISER. I went to many places. I went to 10, 11 places. I don't know which IISERs or which IITs were there. I think these were IISERs. IISERs are newer than IITs.
There is one professor in CSIR IICT Hyderabad. He is working on it. Like during one workshop, he showed his that model.
Yeah, so it is, but I have not been to Hyderabad, in any place in Hyderabad, but I went to some of the IISER, and in that lab everything was done robotically. The compounds was purified by the machine, synthesis was carried out, everything. There was just barely any human interaction. There is a big company where I live, 1 hour away from that. You might have heard of Eli Lilly. It's a world famous company. So there they are saying we don't hire synthetic organic chemists. It's all done by robots. There was a time when I was a synthetic organic chemist by training from PhD, and they don't hire them because it's all robotic now. So we used to spend hours and hours doing column. This person at ISR, he said, who does column now? The machine does it for you. And those who are organic chemists by training, oh my God, it's a lot of sweat purifying by column. It still gives me nightmare, like you are so patiently doing it and you increase the polarity, impurities come together. If you have done organic synthesis, you know that it is so painful, right? So, but basically this is where the science is going, okay? So, for AI to work, you have to feed in data. If you don't feed in data, so that is why that all of these things add up to that one. And all the omics data, genomics, epigenomics, transcriptomics, proteomics, metabolomics, they all have to add. Remember, anytime it is omic means global, across the board, right? And all of this thing for each of us has to be fed in the AI, okay? And then they can actually give you the exact drug for that person. Okay, the more targets you hit, the higher the chance of side effect. So, using AI to explicitly filter out anti-targets. Now, I'm going to show you some case studies. So, imatinib and dasatinib. Dasatinib is just a next generation imatinib. So, when imatinib is Gleevec. When Gleevec was given, it was a huge hit, but then patients developed resistance very quickly. So then dasatinib came into picture. So this is the parental compound, this is the next generation compound. So this was designed to target BCR-ABL, c-KIT and PDGFR. This accidentally polypharmacology allows it to treat gastrointestinal stoma. So this was accidental. It was, people did not know about polypharmacology at that time. This was just a really good luck there. But dasatinib was deliberately made it dirtier, so it inhibits BCR-ABL, SARC, c-KIT, and ephrin receptors. So when people developed resistance towards Gleevec, this drug was much better. Okay, this is a real thing. So remember we were saying PD-L1 inhibitor or angiogenesis inhibitor. This one, sunitinib, this inhibits so many targets at once. Remember, angiogenesis inhibitors receptors are VEGF receptors. So it hits many VEGF receptors, and these are the ones which cause tumor growth, cell proliferation. So sunitinib hits so many targets at once. So it is inhibiting tumor growth plus angiogenesis. It's not a PD-1, PD-L1, but it is inhibiting tumor growth and angiogenesis. So this is a very, very powerful drug, sunitinib. Then lenvatinib. Lenvatinib, look at that, so many of them, it is increasing. Okay, so it is inhibiting cancer, cell growth as well as angiogenesis. So look at that, so many of them at once. Then sorafenib, it inhibits RAF kinases, VEGF, BTGFR, it's really good. Anytime you hear VEGF, that means angiogenesis. VEGF receptor is the receptor for angiogenesis. Okay. So these are some examples. So these drugs and how many targets they are hitting. So they are hitting several of them, and they are extremely effective drugs. One pill, so many things. Okay. Then I was telling you about schizophrenia, right? So there is this drug clozapine. Look at that, how many of them. It's unimaginable, and that is really a game changer for this disease. Brain is really complex, right? This morning I was working on a lecture 30th. I had prepared it yesterday, but I keep on changing it till the last minute every single time. So I was adding something and I was talking to my niece, and she's in class 12th, 11th, 12th. She's going to apply for college and she has to write an essay, and she was telling me that she wants to be a doctor in neuroscience, and she said she wants to write an essay on neuroscience and AI. And she told me some really neat ideas. I have never thought of those ideas. And she brought AI, and I was working on AI as she, like I was putting things on the AI. So I said, I will send you my presentation, but it is cancer in AI, so you read about it. But she gave me some really nice ideas about her own thing, spatial, like, the brain is three-dimensional, right? So how memories are stored, how do we see space, how do we see the depth versus this, and how we are moving hand in three dimensions. And when somebody has prosthetic arm, you think about it, and your arm based on your thinking. So those kind of things telling me how you can use AI to do that. So it's just imaginable the kind of things you can do with AI. Okay. So obviously, when drug is clean, it is much better to get FDA approved. But now FDA, the dirtier drug can be, agencies are adapting. Okay, if a dirty drug gives lives in a country where clean drugs fails, it gets approved. Okay. So AI and multi-specific. Okay, so we will move from discovering accidental polypharmacology to engineering. So Gleevec is accidental. I have nothing to do with Gleevec but I joined when Gleevec has approved, and as part of the kinase group, I got the perks of the approval of Gleevec without Gleevec. There are huge solutions for Gleevec, and these are parties. These were taking people to Mallorca and taking a Mediterranean where they were serving you finger food, and while your ship was going to the Mediterranean. It's the most beautiful part of Spain, the Mediterranean coast. So I have the things I did not at all to Iraq. Okay, but I was just at the right time to.
The branding you're doing for.
Yes, yes. Okay, so, and we can obviously Protax. Glues are something similar to Protax. Protax, in very Protax have a link between, but if you remember those two things are together, that's called molecular. That's, so this is becoming very popular. Digital avatars, digital, so there, right? So they do proteomics, genomics, metabolomics, transcripts, everything. All my history, everything is there. So they know. Then they create a virtual meeting with the exact PGA genetics. They know drugs will, and they will test the drug virtually to what kind of interaction you have, and then they will grade so that they can predict everything. Okay, so this is personalized. This is not vision. This is personalized. Is equal to 1. 1 person, 100 personalized. 3D polypil modulation. Polypharmacology is not lack of precision, it is precision applied to the reality of biological complexity. I hope you understand the meaning of this thing. Precision means you're precisely targeting, but polypharmacology, you can say it is not, it is going to targets. No, it is extremely precise in choosing all targets at once, because biological systems are very complex. You cannot just one target. If you have to change a nation, you have to, do it in many different ways. Okay.